We're now going to leave behind observational studies and move on to talk about the so-called experimental studies. But before we roll up our sleeves and jump into talking about the experimental studies, I want to now review with you what we mean by the phases of clinical trials. Before any drug gets approved by the Food and Drug Administration, it must go through several phases of clinical trials to generate the data that will be used to make a decision about that drug's approval. You're expected on your exam to know what happens at each one of these phases, at each one of the key stages, that is, of the clinical trials. You're supposed to know that in a phase 1 clinical trial, there are no patients at all, nothing but healthy volunteers. A phase 1 clinical trial is the first time that the drug is being given to actual people. Before this, it might be given to animals of one kind or another, but now we're crossing the barrier into giving the drug to people. What we want to do in a phase 1 clinical trial is focus on safety. And the reason we use healthy volunteers is if there's some negative consequences to taking the drug, we think healthy volunteers, not being as vulnerable, are more likely to survive than people that are compromised by an illness or disease. Phase 1 is not about patients. That means a test question where a patient is demanding to be enrolled in a phase 1 clinical trial, the answer is obvious. It simply doesn't happen. Phase 2. A phase 2 clinical trial, importantly, is the first time we're actually giving the drug to real patients. Now we're moving away from healthy volunteers to actual people with the disease who might who might generate some benefit from that particular disease. We're going to get some tangential information about efficacy in a phase 2 clinical trial, but the main purpose here is to get a sense of protocol and dose levels. Simply, how should we give the drug? How often should we give it? How much should we give? Protocol and dose levels. What we're going to do is give the drug a number of different ways, a number of different dosing to a number of different groups of patients, and the patients that seem to have the best response, either by a clear clinical outcome or some sort of intermediate indicator, will decide that's the best way to do the administration of the drug. A phase 2 clinical trial is important in that it is, once again, the first time the drug is given to patients. But remember, we're not really getting information here about efficacy. We're really trying to find out how to administer the drug in order to maybe get efficacy. A phase 3 clinical trial is the main event. In a phase 3 clinical trial, we're now testing, 1, efficacy, does the drug work, and 2, we're looking for side effects. What might be the negative concomitants that go along with the positive benefits? Phase 3 clinical trial is the time when we're really getting the data that tells us at the end of the day, thumbs up, the drug's good, or thumbs down, it doesn't work, or there's too much harm that's done. If a phase 2 clinical trial involves hundreds of patients, a phase 3 clinical trial involves thousands of patients. See, in a phase 3 clinical trial, we have to have a large enough cadre of patients so that side effects can be seen if they in fact are present. Remembering that many of the side effects you read about are, 1 per thousand, 4 per thousand? And so if I only had 100 patients in the study, I might never see those side effects, even though they're in fact present. I therefore talk about thousands of patients in a phase 3 clinical trial because I need to have a wide enough group in order to see the side effects if they are present. We're going to get information about how well the drug works. We're going to get information about possible negative consequences. And those 2 pieces of information are the meat that go into the Food and Drug Administration, the FDA, and they'll make decisions about whether or not the drug can be prescribed by physicians. Now, this journey through the 3 phases of clinical trials is long, involved, and frankly, quite costly. For every 100 drugs that start through these 3 phases of clinical trials, 1 will be eventually approved by the Food and Drug Administration. For every 5 drugs that get significant phase 3 results, 1 will be eventually approved by the Food and Drug Administration. This is a long, costly enterprise where many are tried, but frankly, few are chosen. After a phase 3 clinical trial and after FDA approval, the drug is now out in current usage. But we don't stop watching because, well, we do something called a post-marketing survey. Now, some of you might have heard this called a phase 4 clinical trial, and that's actually a misnomer. It's not really a good name. Post-marketing survey is better because it's what we're doing. We're surveying to see what happens now that the drug is out in common usage. By recent FDA decision, this will have to happen formally 18 months after the drug is out, but frankly, we do this on an ongoing basis to make sure there's no well, unknown detected side effects that we didn't see before. Post-marketing survey is a key part of the enterprise, but it's not a full-fledged clinical trial. We're simply observing what happens to people taking the drug. We will and will always certainly continue to find drugs that look good in the phase 3 clinical trials being, well, having too much negative consequences for the patient in the post-marketing survey. And the reason for this is there may be some serious side effects that are very, very serious but relatively rare. Let's say we have a drug that kills 1 in every 100 patients that take it. Well, if I had 3,000 patients in my phase 3 clinical trial, I might have never seen that side effect. But once I have the drug out in common usage and maybe 40 million people are taking it a year, I'm killing off 40 people a year. I have to have a large enough group of people for some of these rare but very serious side effects to appear. When we do find serious side effects, there are a number of options that can be taken. One of the most common is we simply pull the drug off the market. Usually the person who put the drug on the market voluntarily withdraws it from the market. The most talked about current historical issue is Vioxx. Vioxx was pulled from the market because we discovered it does have people, well, increase the risk, I should say, of people getting certain kinds of heart attacks and heart disease. Other drugs, nifazodone, no longer on the market because of, well, liver toxicity. Nifazodone was a useful antidepressant, but it's no longer available to you. Again, we're going to find drugs that have these side effects, and even if we tried hard, we couldn't have anticipated it before because these side effects, although severe, are relatively rare. The other option we have is we sometimes leave the drugs on the market but put down what's called a black box warning. A black box warning is simply a warning with, this will stun you, a black box around it to make sure people see it so it stands out. We put black box warnings on some of our antidepressant drugs, paroxetine and other SSRIs, as they're being given to adolescents, but increasingly we realize this is also an issue for adults. Certain of the antidepressants, in fact many of them, are at least for the initial period when people are on the drug, likely to heighten the chance of somebody committing suicide. You're expected to know what happens at each one of the 3 phases of clinical trials and to have some familiarity with what happens in the monitoring phase, the post-marketing survey, after the drug is available and on the market. With that, let's roll up our sleeves and launch right into a discussion about what we mean by clinical trials. Now, I laid out observational studies for you as a sequence, and that's the right metaphor. But it's not really the way to think about clinical trials. The way to think about clinical trials is not a stair-step sequence, but as different pieces that you cobble together in order to construct the kind of clinical trial you yourself want to have. My son plays with something called Legos. You know what I'm talking about? Little plastic blocks you snap together, build whatever toy you want to play with. Well, that's really the right metaphor for thinking about clinical trials. They're pieces you assemble to construct the particular kind of trial you want to have. What we want to do is review what the pieces are, and then in your exam you're going to have to pick all the pieces that describe the study that you're presented with. The biggest piece of the clinical trials, the hugest Lego block in the set, is the RCT, Randomized Controlled Clinical Trial. RCT is the core on which we're going to build with all the other components that are available to us. Let's take a moment and walk our way through some of the words here in this randomized controlled clinical trial. First, let's talk about the word controlled. You want to know, briefly, how a clinician thinks differently than a researcher? It's easily described. A clinician gives a patient a drug and asks the simple question, does the patient get better? A researcher gives a patient a drug and asks the question, does the patient get better relative to the comparison group? See, research is the art of comparison. What we're trying to do is not just see if the drug works, but how does it work relative to the baseline? And the control group is our comparative baseline. There's 2 basic kinds of control groups that you find. One is a placebo group. A placebo group is a group that gets absolutely everything that the intervention group gets. They get the same attention, the same interaction, the same analysis, the same contact with the research personnel. The only thing they lack is the magic ingredient in whatever pill they're taking. Now they're going to get the same size and shape of pill. You're not going to be able to tell from what they're taking what they're getting. It won't have what we believe to be beneficial pharmacology. It will be some inert substance. We often talk about using milk sugar as a placebo. What I want you to hear from that description, therefore, is not, in a placebo study, the the patient gets nothing, because if you listen to what I told you, in a placebo group, the patient gets absolutely everything the treatment group gets, except they don't get the beneficial pharmacology. What that tells you, therefore, is what we're able to answer in a placebo control study is a simple question. Does the giving the drug work better than not getting the drug? You have to know that many patients get better in the placebo condition. Some generalized analyses of available research show that often as much as 40% of patients get better in the placebo condition. Placebo doesn't mean the patient's getting nothing. It means they're not getting what we believe is the new beneficial pharmacology. You also should be aware that when we do PET scans of the heads of patients in placebo conditions, we find noticeable, and by this point, predictable neurological changes in the brain. Placebo, once again, doesn't mean the patient's getting nothing. It means the patient's getting everything, but they're not getting the new pharmacology. But sometimes, not giving anything that we know is beneficial to this treatment group or this research group can be, well, unethical. I mean, suppose I'm looking for a new drug to treat patients who suffer from cancer. Well, to give a new drug versus a placebo would be absolutely unethical because we know the cancer untreated will simply spread. In these circumstances, we use a different research design. Instead of a placebo group, we use a standard of care group. In a standard of care group, we're comparing the new treatment to the current treatment. And by that, we're answering a different question. Not, does this treatment work better than not giving a new treatment, but does this new treatment work better than the old treatment? Notice, in a standard of care design, you could discover that the new treatment doesn't work any better than the old treatment, but it might well be the case that the new treatment is actually very beneficial relative to a placebo group. But we'll never know because withholding treatment to the cancer patient would be simply unethical. We won't do that research. Whether it's placebo or standard of care group, the art of experimental research is to have a control group, a comparison group, so you can say not just the drug works better, but the drug works better than. And the than is defined by one of these two kinds of control groups. The second word I want to draw your attention to in this randomized controlled clinical trial, RCT, is the word randomized. In a well-run clinical trial, whether or not patients end up in the treatment or the control group has to be the result of a random process. Not subject self-selection, no. Not the whim of the researcher making the assignment. There has to be some random, impersonal process that puts people into one or the other group. Most common way this is done is with a table of random numbers or maybe generating random numbers out of a computer, where all the even odd numbered people go to one group, all the odd numbered people go to another group. It's absolutely essential that you have random assignment because each individual is different. We have to make sure that the reason we find differences between the two groups in our study is not because we have different kinds of people, but those differences are evened out. And so we can have the baseline of evenness so we can see the difference due to the drugs. The whole point about random assignment is to equalize out individual differences so they occur with the same frequency in each one of the groups. That provides the baseline, the backdrop, if you will, against which we can see the effects of the drug or intervention. Random assignment is a key, crucial feature in every randomized controlled clinical trial. Okay, with that major block in place, let's talk about other things that we can cobble on and clip to the block. The first of these, already known to most of you, I'm sure, is a double-blind design. Double-blind design means, one, the patients don't know if they're treatment or placebo, because, come on, if they knew, it's not much of a placebo. And two, the researchers who interact with them can't know either. The reason the researchers can't know is twofold. First, if the researchers know, they might slip up and inadvertently reveal that to the subjects in the study. And that wouldn't be blinded. The subjects would know if they're getting treatment or placebo. But secondly, and more importantly, researchers might act differently to people if they know they're getting the drug than they act to people that are not getting the drug. And just that different action could distort or skew the research results you get. If you do this correctly, nobody that has contact with the subject knows whether the subjects are in the treatment or the placebo condition. Now, clearly someplace somebody knows. used as a laboratory technician or computer jockey who has the code list saying who's in what group, but this is someone who never has contact with the subjects. Somebody once described to me a double-blind design where they said the only person that knows who's getting the real drug or not is the nurse who hands out the medication. Well, we all understand that's not a double-blind design because the nurse knows and she has interaction with the subjects, and that could lead to distortion of the results just by that knowledge that she carries. Every phase 3 clinical trial worth its salt is a double-blind design. You can pretty much assume when you see phase 3 that it is double-blind. All right, here's the key exam issue. Never break the code in a double-blind study unless you're closing down the whole study. Because once you break the code for one patient, you've literally broken it for everybody. Never break the code unless you're closing down the study. The questions in the exam pool where a patient, a family member, a hospital administrator, a government official, I love that one, come to you and say, let me know which drug this person is getting. When you see that question, remember me talking to you right now, never break the code unless you're closing down the study. Why? Because when you break the code, you've closed the study. See, all I know as a researcher in a double-blind design is I have group A and group B, but I don't know which group is getting the drug and which group is getting a placebo. But once I know this person in group A is getting the drug, I now know everybody in group A is getting the drug, and the double-blind design has collapsed. We have a double-blind design because we need one. Never break the code unless you're closing the study. Next, let's talk our way through what we mean by community trial, and we'll talk about this relatively briefly. A community trial is merely looking at how well an intervention works in the real world. Out in the real world where real people live real lives. Not in the controlled confines of the hospital or laboratory, but out where real people make real life decisions. Want to see how it works when people are doing lifestyle changes. Want to see how it works when people are taking drugs that might interact with things in their life or things in their environment. Community trials are essential to nail down the knowledge we need to understand this process. Because remember, what's important for medicine is not what works theoretically, but what works actually. If it's theoretically a good drug, but in usage it doesn't have benefit, we don't care and will simply stop prescribing. More important for your exam is to understand what we mean by a crossover design study. Now please note the terms here. Crossover study. A cross-sectional study, we talked about that previously, that's from, observational studies. A crossover study is a kind of experimental study that is a clinical trial. In a crossover study, we're trying to find a way to solve the sometimes conflicting demands of research methodology and research ethics. Research methodology requires something simple out of you. You give the drug to one group, you don't give it to the other group, and you can simply compare the results. That's easy. But suppose it's a drug that doesn't cure the patient outright, but instead you think might help them live, maybe 2 years longer than if they're not on the drug. Well, if I give it to one group and don't give it to the other group, isn't this group that didn't get the drug, if it turns out the drug was effective, going to be really mad and angry at me? So, in order to make sure we haven't withheld potential benefit, we have to give the drug to everybody. Well, wait, if I give the drug to everybody, then I have no research because I have no comparison. I can't say the drug works better than because there is no comparison group. Everyone's getting the drug. Into this mix comes our solution, the crossover design study. Crossover design studies can be very complex, but at root, they are very simple. The beginning of this, listen, always double-blind design. We have Group A and Group B. Unbeknownst to them, Group A is getting the real drug while Group B is getting a placebo. And then at some predetermined, pre-wired point, crossover occurs. Now Group A goes over to get the placebo, and Group B goes over to get the real McCoy drug. Notice what we have here. First, at every step of the process, we have a comparison group. Every step of the process, we have somebody on and off the drug, so comparison is possible. And please also notice, we can also compare within groups of subjects. I can see how A did on the drug and off the drug. I can see how B did off the drug and on the drug. A nice tight comparison. Notice what else is true. I have no untreated study participants. Everybody in the crossover design study gets the drug at some point. Yes, A gets it early. Yes, B gets it late, but everybody gets it. And so if there's any benefit to be derived, each of these subjects will benefit from whatever good there is to be gotten. In addition to solving our ethical research methodology conflict, a crossover design study has another advantage. It's simply easy to recruit subjects to participate in this study. If you're trying to recruit subjects to participate in a standard placebo study, you have to tell them, informed consent being what it is, that please sign up for the study and you'll either get the new drug or you'll get the placebo. And patients universally don't understand this. They say, well, I don't want the placebo. Give me the new drug. You explain how the research works and they say, yeah, somebody else can have the placebo. I want the real drug. You have to tell them maybe you'll get the real drug, maybe you won't. I won't know in a placebo group study. In a crossover design study, you can tell every patient and tell them honestly, please sign up for my study and I guarantee you'll get the drug. You can't say when they'll get it, but you can be sure you'll guarantee they'll get it. And that's an easier conversation to have with your patients. They feel much better about going forward and much better about participating in the study. We started doing crossover design studies very heavily as we're looking for, well, treatments for patients that have full-blown AIDS or are simply HIV positive. We know that AZT is an effective agent because of the results of a crossover design study. But wait, AZT is not standard of care for HIV anymore, right? Standard of care is now a protease inhibitor drug cocktail, now available in a single pill. But you ought to know the research on the protease inhibitor drug cocktail never finished, never completed. We never formally completed the research. Do you know why? Well, in this double-blind study, in one arm of the study, we didn't know which one, 2 people died. In the other arm of the study, we didn't know which one, 18 people died. If you're confronted with that on the exam, what action will you take? Well, that's obvious. Close the study. And the issue, please note, is not that people died, it's the differential mortality. If I had 18 and 18 die, we'd keep the study going. But when I have 2 versus 18, your guts ought to be screaming, wait a minute, something's wrong here, and we pull the plug and the study design gets closed. Because first, do no harm is not just a dictum for medical practice, it's also your directive for all aspects of medicine, including medical research. If you think there's harm, you close the study. And those are a run through the different pieces that we use to construct our experimental studies. The big core piece is the RCT, randomized controlled clinical trial. We like to do, if at all possible, a double-blind design along with that. And then, if we have to, a community trial, or if we have to, a crossover design. The final comment I want to make to you here is don't always think that a crossover design study is best, because the rule for research is actually simple is best. Occam's razor applies here as it does in so many areas of life. The simpler design is the better design. If you want to get a direct answer, you ask a direct question in order to give you the cleanest answer possible.